So why is GFP fluorescent? Well, it's quite unique amongst proteins in that it does emit light in the visible regions. Most proteins can actually absorb light, but they do it in the UV region. But GFP can do it in the visible region. And this is because it forms what we call a self-encoded chromophore. So what it does, it takes its normal amino acid sequence and rearranges 3 contiguous residues to form a new chemical structure. And this new chemical structure is para-hydroxybenzylidene imidazolinone. Not even I can say it properly. But that is essentially the difference between normal proteins and GFP. And this is a post-translational modification event, and it happens once the protein has been produced, and in fact when it folds up into its native structure. Because the chromophore itself is buried in the middle of what we call a beta-barrel protein scaffold. In other words, the protein structure looks like a barrel, and it's formed primarily this barrel structure of the beta strands. And there are 11 of them which form that barrel structure. And those 3 amino acids, which undergo covalent rearrangement, are buried right in the heart of that barrel with a helix that goes right through the middle. But that chromophore is still part of the whole polypeptide structure. It's still part of the whole amino acid sequence. So there's no break in the structure. And normally those 3 amino acids will comprise of a residue, followed by a tyrosine, followed by a glycine. Now the tyrosine and glycine are generally conserved amongst fluorescent proteins, whereas that first residue of the 3 can vary. So if you look at the structure of the chromophore itself, we can see where the name sort of comes from and why it's named in such a way. Because we have something called the para-hydroxybenzyl component, or probably better termed as the phenol or P-ring component. That comes originally from the tyrosine residue of the original amino acid sequence. Then you've got this idine part of the name, or which is what we commonly refer to as the methylene bridge, which connects the P ring to the next ring component, or the imidazolinone ring, or I ring. And so we can see how the breakdown of that name comes to the different chemical components. Now the I ring is comprised of the first and third residues. And the only thing that's needed to make this new chemical structure is a folded protein and molecular oxygen. Molecular oxygen is absolutely essential. We don't get that full chemical structure formed unless there's oxygen present in the system. But that chromophore is absolutely essential. Well, we call that chemical structure a chromophore, but that chromophore is absolutely essential for the function. But the rest of the protein as well plays a key role in defining the fluorescent properties of GFP. So the structure as a whole should be taken into account, because if that beta barrel structure doesn't form, first of all, we don't get the chromophore formed, and secondly, if even if the chromophore is formed and then that beta barrel breaks apart, fluorescence is lost. The chemical structure of the chromophore is there, but because it's exposed to water, it no longer emits the light out. If it absorbs blue light, instead of emitting that back out as light, in green light, it will actually dissipate its energy through the system in a thermal way, so just as heat. But the key thing is, if you look at that picture on the right there, you look at all the interactions made of the chromophore with the rest of the protein and with water molecules. And in fact, this is what fine-tunes the fluorescent properties. And all these interactions are so important that we can actually go in and tinker with the fluorescent properties of GFP and make them better, or make them have different color outputs or respond to different color inputs based upon how we tinker those different interactions by a process called protein engineering.